You're listening to the Research Report Show
on Varsity Podcasts. Hello, everybody, and welcome to yet another exciting episode of the Research Report Show. My name is Julian, and today in the studio, I am joined by Michelle Ngwarai, who is a Master's in Medical Microbiology at the University of Cape Town. Welcome to the show, Michelle.
Thank you, Julian.
Okay, so Michelle, looking at the abstracts that you sent to me, the title of your thesis is centered around the stool, microbiota, and infant wheezing, which my understanding is looking at bacteria found in the stool of children and how that correlates with infant wheezing. But before we dive headfirst into that, Michelle, do you want to give us a background about where you're coming from and how you got to this point of doing your master's?
Cool. Thank you. So I did my undergraduate degree in biochemistry and microbiology, and I almost immediately fell in love with biochemistry. I then went on to do honors in molecular and cell biology in the same department. And for my honors project, the main focus was resurrection plants. And it was during this time that I realized I missed that microbiology aspect of it. So looking at bacteria and analyzing their effects and so forth. So I made sure that my master's would be in microbiology. So I applied for microbiology, medical microbiology at the health sciences faculty. And that's what I'm doing right now.
Okay. You said something that was interesting about your honors. You mentioned resurrection plants. This episode is not about your honors, but just out of curiosity, are resurrection plants that die and then they come alive again?
That's close enough.
Like a phoenix?
Not a phoenix, because a phoenix has to completely die and then whatever happens and then it comes back to life. But these are plants that are able to survive with as little as 5% of relative water content. So when you look at them, they have literally no life in them. They're not green. They're brown and they're dry and brittle. And you would think that even if you would water such a plant, you won't be able to get any life out of it. But if you water these plants, within 24 hours, they'll be green again. They'll be making chlorophyll and making food and even flowering after a couple of days.
All right, cool. That's interesting. I didn't know that there were plants that could come alive in literally 24 hours. Another burning question just at the back of my head before we dive into what your current master's research is about is, what is the difference between biochemistry and microbiology? Just a layman's overview definition, for the ordinary man like myself or any of our listeners who might be asking the same question.
Okay. So, biochemistry is a mix of biology and chemistry. So, which is a lot to do with chemicals and biological chemicals, what they do in for example, the human body, what they do in an animal body, what they do in bacteria as well, and how those effects come about relating to physiology. And microbiology, micro means small, like very small. Yeah. And then biology, so it's looking at the biology of very small life forms.
Okay.
To say the least.
Okay, cool. So let's dive straight into your master's. What is the exact topic for your master's research, like your thesis? What would be the topic of that?
So my topic is the stool microbiota and infant wheezing in the Drakenstein region, South Africa, Cape Town.
Okay. Do you want to unpack that?
So, as you said before, it's looking at the bacteria that's present in the stool in a longitudinal period of these infants from birth till the age of 2 years, and relating that to whether or not they develop wheezing. And we're looking at wheezing as the outcome in this because it's shown to be one of the precursors of asthma later on in life. So at the age of 2 years, you can't determine whether a child has asthma or not, but wheezing is a precursor of asthma usually. So most children that develop wheezing that early on in life are then going to go on and develop asthma. So that's the outcome that we're looking at.
Okay. And how far are you into this master's program? How has the journey been so far?
So comparing with undergrad and honors, It's been a long journey. It's been an eventful one. It's been partly rewarding. I've learned so much to do with data analysis, to do with sifting and working through Excel to pick a cohort that I want. So a cohort is the group of participants that I'll be working with. And I've learned how to do this. I've gone on a couple of online and training courses to learn how to analyze the data that I have now, as well as the data that I'm going to have as soon as I'm done with my laboratory work. And to answer the question of what I've done so far, so I've done a lot of pre-data analysis, if you can call it that, looking at my data, looking at the data that's available, because this is a prospective cohort. So it means they collect samples way before the study question or the many study questions within the main study have been formulated. Okay. And then, so my supervisor came up with the question, and then we look at the data that's available and see what samples do we have compared to what we want. And with research, you almost have an ideal, but it's hard to reach that ideal usually. So you have to work with the data and see, okay, if we can't have 10 samples from every individual, how many samples can we get? And then it's a lot of that. And then now I'm doing a bit of laboratory work. So that involves DNA extraction of these samples on a manual platform. And I'm going to be doing the library prep and then carrying on to sequence these samples to see what specific bacteria are in these samples. And then when we're done with the sequencing, we'll then marry that data with the clinical data, which are some of the variables that we're looking at. So do these children have siblings? Do these children stay in a apartment, in a house? Do they have pets? And then we're able to see from the clinical data what type of children are developing wheezing. And can we see a clear difference between our cases, which are the children that develop wheezing, and the controls, which are those that don't develop wheezing?
I think you lost me a bit there when you were talking about clinical data and then marrying that with the other data. I guess my assumption is that you mentioned that you're looking at stool samples of, say, 0 to 2 years old.
Yes.
And those stool samples come with some metadata associated with them. Is that the clinical data?
Yes, that's what we refer to as the clinical data.
Okay, and then the other data, is that the results of the lab work?
Yes.
Okay, so what you then do after you've done the lab work on the stool samples is marry the results data from that with the metadata that you already had for the stool samples and then draw conclusions from that.
Yes.
Okay, cool. So how long is this master's program, and how far along that spectrum are you? So if it's a 2-year program, how far in are you, or is it longer than 2 years?
So ideally, the master's program that I'm doing is supposed to be 2 years. And this is the end of my second year, but I'm not quite finished yet. So I still have a couple of months that I'm going to be finishing off my laboratory work, my analysis, as well as writing up.
And from taking away from the lab work that you've done, you mentioned that there's some data analysis that you would do. What sort of tools and maybe techniques do you use for this data analysis? So
I'm in no way a pro at the data analysis yet. It's a lot of coding. So it involves a program called R.
R being the statistics language.
Yes.
Okay.
Sorry. Yes. Statistics language called R. And we have a statistician actually who is very kind enough to help us during this process. And she writes the scripts. So what we do is we talk to her and we tell her, this is the data that we have and this is the question that we want to answer, and this is how we want to represent the data. Okay. So she then creates a script, and it's a lot of back and forth process of manipulating slightly or adding something else, removing something else until we get the perfect story that we want to create using our results.
Okay. Are you able to make any conclusions as yet, given the data that you've looked at, or that you would say is still a work in progress?
It's still very much a work in progress because I haven't gotten to that analysis or the stage where we marry the metadata with the results from the lab work.
And once you're done with with this master's, what would be the next step in this sort of research of examining the stool samples of 0 to 2-year-olds to try and determine how that relates to infant wheezing? What is the next step once you've got a set of results or conclusion from this study? Will your output be the input to someone else's research, or maybe your own research if you're carrying on with like a PhD or something along those lines?
So microbiome data or microbiome studies, which is what this study is, they're very interesting topics, and it's very much still growing. So a lot of the things that I'm doing right now in the current setting are very much at the basal level or foundation, if you may call it that, because there hasn't been that type of study that has been carried out in Africa and in South Africa. Okay. So that's one of the main reasons why we're doing this study. There have been a lot of studies carried out, say, for example, in the States, in England and Europe, and so many other countries. So having that done in our setting is just the base, and then it'll be good to always try repeat the same type of study as I've done. So pretty much getting more participants than we have. So one of the problems with this is you don't get to have the number of participants that you want, because you're enrolling people. Sometimes you don't get the thousands or the millions maybe of numbers that you may want that will make your data a bit more significant. Okay. So repeating that study with a lot more participants will be good, and with a lot more sample points that are consistent will be a very good thing to do. But it will definitely be used as a foundation to the next project. So probably even building on from the same project, looking at participants not only from 0 to 2 years old, but from 0 to 5 years old, because this cohort collects samples from birth to 5 years old.
Okay.
Yes.
Okay. That sounds interesting. And personally for you, what is the next step after this? You know, are you already that far ahead looking to what the next step is beyond your master's?
Yes and no, because part of me wants to go to work because I'm interested in diagnostics, if you may call it that. Someone who would work, for example, say at Pathcare or in a hospital or in a lab. So that's doing the processing, pretty much mass processing of samples, analyzing different things for patients or whether it's for research purposes. So I'm interested in that partly. I'm partly also interested in pursuing a PhD, but I'm not necessarily sure if it will be in the same type of field or in something different.
Okay, thank you very much, Michelle. On a lighter note, what do you like to get up to on a weekend? What does Michelle get up to in Cape Town? Or what are some of your interests outside of diagnostics work, you know, working in the lab with samples, working with Excel spreadsheets? What else do you like to do in your spare time?
I love reading. So I would love to, or I really like going to exclusive books and getting myself a new book and just hiding away from everything else and doing that. I also love camping. So it's always nice to be out in the woods and sleep in a tent, hearing all the noises of animals around. Yeah.
Okay, under an African sky.
Yes.
Stars and all that. That's interesting. Thank you very much, Michelle, for joining us in the studio today. I'm sure our listeners would echo the same sentiments that they've learned a thing or two about working in the lab, about correlating stool samples with infant wheezing, and maybe even some of our listeners who might be interested in doing further research might get inspired from listening to this. So thank you very much for that. That's all we have for you today. To our listeners, thank you also for joining in and listening. My name is Julian. I've been on for the Research Report show. We had Michelle, who's a master's student at the University of Cape Town, where she's doing her master's, doing some research that's correlating some bacteria that's found in stool samples with infant wheezing. That's for kids between 0 and 2 years old. So that's all we have for you today. Check out our website, www.varsitypodcasts.co.za for more shows. And until next time, keep well.
This is Varsity Podcast.